Hi class, this is your instructor, Skylar Huff. We're in Chapter 18. This is Genomics. The pace of discovery in biology in the last 30 years has been dramatic. Starting with the isolation of the first genes in the mid-1970s, researchers obtained the first complete genome sequence, that of the bacterium Haemophilus influenza, and that is shown to you here. So it's with genes of similar function have a similar color. Looking closely, I see a number of colors that look to be pretty similar. By the mid-1990s, that was done. So a complete draft sequence of the human genome had been completed by the turn of the 21st century. Put another way, science moved from cloning a single gene to determining the sequence of 1 million base pairs in 20 years, to determining the sequence of 3 billion base pairs in 5 more years, to now being able to sequence 20 billion base pairs in a few hours for as little as $1,000. In the last chapter, you learned about biotechnology. Here, you all will see how some of those technologies have been applied to the analysis of whole genomes. Genomics integrates classical and molecular genetics with biotechnology to study the structure, function, and relatedness of genomes. So in this chapter, we won't do it all, we'll do some. Let us begin. We use maps to find locations, and depending on the nature of the location, we use different types of maps. A map of the world, a map of a country, or street maps of a town. In genomics, a gene can be located to a chromosome, to a sub-region of a chromosome, and finally to a precise location in the sequence of chromosomal DNA. A map at the DNA sequence level requires knowing the entire sequence of the genome, once technologically possible. So knowing the entire sequence of a genome is useless, however, without knowledge of how a genetic region affects particular phenotypes. This sort of information resides in genetic maps. To make an analogy from the geographic maps, finding a single gene within the sequence of the genome is like trying to find a house on a map of the world. So we construct different kinds of maps of genomes from different kinds of data and for different kinds of analysis. Fundamentally, there are 2 kinds of maps, genetic maps and physical maps. Both these types of maps use genetic markers, which can be any detectable differences between individuals. Genetic maps are abstract maps that place the relative location of genetic markers on chromosomes based on recombination frequency. Physical maps precisely position genetic markers in the genome, with the ultimate physical map being the complete DNA sequence of a genome. The field of genomics unifies many different types of maps or views of the genome to facilitate large-scale analysis. Although not technically the study of a genome, the relationship between the genome and the express RNAs and between the genome and the express proteins can be studied. Genomic studies have shed light on evolutionary relationships, genetic susceptibility to disease, and development. So classical genetic mapping uses crosses to determine the number of recombinant progeny. In humans, use pedigree information and statistical analysis. So distances measured in centimorgans, where 1 centimorgan represents a 1% recombination frequency. Genetic maps locate genetic traits on a chromosome, but have limitations. So distribution of crossover is not random. Recombination frequencies vary between individuals, and relationship between genetic distance and physical distance varies across the genome. To physical maps. So there are 3 main types of physical maps. Restriction maps made using restriction endonucleases. Chromosome maps. And then thirdly, sequence-tagged site maps. So distances are measured in base pairs. Base pair measurement unit is 1,000 base pairs, or base pair units, or kilobase. And then not necessarily no DNA sequence. Restriction maps are generally used for DNA molecules less than 50 kilobases. Cut DNA with restriction enzymes and analyze the pattern of fragments. It can be applied to larger genomes if the DNA is first fragmented. Restriction mapping is then repeated and used to put pieces back together based on size and overlap into a contig, a contiguous segment of the genome. So this is showing you how restriction enzymes can be used to create a physical map. By chromosome mapping, whole genome can be mapped at low resolution using stains to produce visible bands. So you identify chromosomes and divide it into regions. So each chromosome has a P and Q arm and further broken down into subregions by banding. So fluorescent in situ hybridization, or FISH, can be used, and then mapping the resolution up to 25 kilobases. So FISH correlates clone DNA with cytological maps. So the karyotype of human chromosomes showing the translocation between chromosomes 9 and 22. You see it right there. So that's a 9-22 translocation. And then FISH using the BCR, green, and the ABL probe, which is red, as you see there at the bottom. So the yellow color indicates fused genes. And then nextly, the ABL gene and the fused BCR-ABL gene both encode a tyrosine kinase, but the fused gene is always expressed. So we have the sequence tagged site. So they're a short, unique stretch of genomic DNA that can be amplified by PCR. So the sequence tag site mapping asks whether two sequence tag sites are on the same DNA molecule. And I wish DNA was together, you all. So DNA is randomly fragmented. If two sequence tag sites markers are found together on fragments of DNA, then they are relatively close together. So sequence tag sites provide a scaffolding for assembling genome sequences. And there you're seeing it there. So this is creating a physical map with sequence tagged sites. Sequencing genomes. The ultimate physical map is the base pair sequence of the entire genome. All sequencing approaches adapt DNA replication to an in vitro environment. Advances in genomics enabled by newer and faster sequencing technologies. So it uses dideoxynucleotides. So again, there are dideoxynucleotides in DNA sequencing. So they have chain terminators, they are fluorescently labeled, and they are incorporated at random. So the fragments are separated by a capillary tube, electrophoresis, and then the detector reads the sequence to a computer. So we're looking here, getting into automated sequencing. So the sequence to be determined is shown at the top as a template strand for DNA polymerase with a primer attached. In automated sequencing, the ddNTP is labeled with a different color fluorescent dye. And that's the dideoxynucleotide, ddNTP. So that allows the reaction to be done in a single tube. The fragments are generated by the reactions are shown, and when these are electrophoresed in a capillary tube, the laser at the bottom of the tube excites the dyes and will emit a different color that is detected by a photodetector. So now we're getting to next generation sequencing. So new technology vastly increases the speed of sequencing and reduces the cost. Various next generation technologies have similar features. DNA sequence without constructing a genomic library by conventional cloning. Millions of sequencing reactions can be formed simultaneously, and sequencing reactions occur in solution and are read directly. No electrophoresis. So we have Illumina DNA sequencing. So the single nucleotide DNA molecules are attached to a solid surface, and then the DNA molecules are copied using PCR amplification, and that is what is PCR to amplify the template. So challenges here for next generation sequencing. So sequencing many molecules simultaneously, but producing less information per molecule sequenced. Read lengths tend to be short, 50 base pairs up to 1,000 base pairs. It requires more computing power for genome assembly, and technologies that generate longer reads are more error prone. Assembling genome fragments. So clone contig assembly. Large clones are arranged into contiguous sequences by restriction mapping or STS analysis, and the large clones are then fragmented into smaller pieces for sequencing. 2. Shotgun sequencing. So DNA is randomly cut into smaller fragments. It is cloned and then sequenced. Computers put together the overlaps. It does not rely on genetic or physical maps. So the clone contig method, you have the large DNA clones that are first isolated. These are arranged into contiguous sequences based on overlapping tagged sites. Large clones are then fragmented into smaller clones for sequencing, and then the entire sequence is then assembled from the overlapping larger clones. And then the shotgun method: cut the DNA of the entire chromosome into small fragments and clone, and then sequence each segment and arrange based on overlapping nucleotide sequences. The Human Genome Project. So the physical and genetic maps produced by the mid-1990s, the draft sequence of the entire genome was published in June of 2000, and the first reference sequence replaced draft sequence in 2003. It had fewer sequencing errors and gaps and greater coverage. Telomere to telomere genome sequence published in 2022. That took some time and some computing power. So characterizing genomes. The Human Genome Project found fewer genes than expected. Initial estimate was 100,000 genes. The number now appears to be about 20,000 genes. That is not that many. Humans, mice, and pufferfish all have approximately the same size genome, half as many as rice. Complexity of an organism is not necessarily related to its gene number or genome size. And this shows you the relative size and complexity of genome based on organisms you may have heard of and those you may not have heard of. So you see prokaryotes there in blue. So roughly, let's say maybe 9 million base pairs and just under an estimated 10,000 genes. And if you look to the far right-hand corner, you see Homo sapiens near the mouse. You see the Anopheles mosquito near the fruit fly going left. Malaria coming back to the left. What else do we have here? Theocrest, which is Arabidopsis thaliana, used in research all over the world with a lot of genes, ironically. And then if you look down to the middle, just a bit more than the bacteria, we see fission yeasts and Baker's yeasts. Look at that genome. Nonetheless, let's continue. So human genome variation. Work is ongoing to create a pangenome that better reflects human diversity. Our random genome is 99.6% identical to the reference genome. And then currently there are greater than 5 billion single nucleotide variants in human genetic variation database. That is a lot of variants, you all. 5 billion single nucleotide variants. And those are referred to as structural variations as well. So the Wheat Genome Project. I'll be here briefly. So modern wheat, Triticum acetium, gives a better understanding of wheat genomics and will help breeding efforts to increase yield, improve drought, and disease resistance. So the wheat genome is allohexaploid. So there are 3 diploid ancestors. It has 3 distinct genomes, each consisting of about 5 billion base pairs of DNA. And there are 21 chromosomes, 7 from each ancestral genome. The 2 main challenges with the Wheat Genome Project results are the large size, the large amount of repetitive DNA, including nearly identical sequences scattered throughout due to the polyploid nature. In 2017, the third attempt covered about 78% of the genome, but contigs were short, making the assembly a challenge. And then additional sequences produced longer contigs. Genome annotation. So the addition of information to the basic sequence information. So you have the structure and function of the gene, and then attempts to add biologically relevant information to the DNA sequences in the genome. So genes are identified by open reading frames. So algorithms look for those open reading frames. They begin with a start codon, that's AUG, and contain no stop codon for a distance long enough to encode a protein. And they can also look for promoters in that promoter region. Now this is BLAST. I don't know that you all have heard of BLAST. So BLAST is the basic local alignment search tool. So it's a search algorithm used to search the National Center for Biotechnology Information databases, that's NCBI, for homologous sequences. So it permits researchers and people just like you to infer functions for isolated molecular clones. And then by way of bioinformatics. So that is the use of computer programs to search for genes and to assemble and compare genomes. So I hope that this intrigues some of you, because this is where we need more of students like you. So to express potential gene sequences, total mRNA in tissues is isolated and converted to cDNA. So the cDNA is sequenced, then mapped to genomic DNA. So sequencing only the ends of cDNA called express sequence tags. Express sequence tags. So this is another form of STS that can be used or included in physical maps. So now genome organization. Genomes consists of two main regions: the coding DNA, which contains genes that encode proteins, and then the non-coding DNA, regions that do not encode proteins. So non-coding in eukaryotes. Each cell in our body has about 2 meters of DNA stuffed into it. However, only about 2.5 centimeters, approximately 1% of that DNA, is coding sequence. So several major types of non-coding human DNA have been described. So we have non-coding DNA within genes, we have structural DNA, and then we have simple sequence repeats. And that does include short tandem repeats. We have segmental duplications. We have pseudogenes, those are inactive genes, and about 15,000 characterized in the genome. There are microRNA genes. We also have long non-coding RNA. Yes, long non-coding RNA. And now to the transposable elements. So these are otherwise known as transposons. Thanks a lot to McClintock for these. So they are DNA sequences that move within the genome. So they are mobile genetic elements in 4 types. So you have long interspersed elements, short interspersed elements, long terminal repeats, and then dead transposons. The ENCODE project. So this is the Encyclopedia of DNA Elements, and it aims to identify all functional elements in the genome. Primary conclusion is that 80%, or possibly as low as 20% of the DNA sequences in the genome is functional. Of that 62% is transcriptionally active, although most transcribed RNAs have no known function. And that's by way of the ENCODE product. A functional element is a DNA sequence that results in protein production, is transcribed, or has a distinct reproducible biochemical nature. So it's a bit of intrigue here. So some have criticized ENCODE's definition. So it's not necessarily synonymous with biological function. In evolutionary biology, the selected effect definition of the function requires that the function of a characteristic is the one selected by purifying selection. Some argue that only DNA sequences under purifying selection should be considered functional. And then comparative studies suggest that approximately 5 to 15% of genome is functional under this definition. So now to comparative genomics, and we're nearly done, you all. So comparative genomics uses information from one genome to learn about another genome. For example, over half of Drosophila genes, that's from a fly, the fruit fly, have human counterparts. Synteny refers to the conserved arrangements of DNA segments in related genomes, and it can obtain information about an unsequenced genome by comparing it to a sequenced or a syntenic segment of another genome. And this shows grain genome rearrangements of similar chromosome segments. Shades of the same color represent species of DNA that are conserved among different species but have been rearranged. By splitting the individual chromosomes of major grass species into segments and rearranging the segments, researchers have found that the genomic components of rice, sugarcane, corn, and wheat are highly conserved. This implies that the order of the segments in the ancestral grass genome has been rearranged by recombination as the grasses have evolved. Functional genomics. So functional genomics is the study of the function of genes and their products. It uses high-throughput techniques to learn about the products of a genome. 3 separate but related categories are study of all the RNA molecules produced by a genome, or that transcriptome, study of proteins produced from the genome, the proteome, and the study of the interactions made between proteins. Back to DNA microarrays. DNA microarrays are gene chips. They enable the analysis of gene expression at the whole genome level. And this is what I kind of got to in the last chapter. DNA fragments are deposited on a slide, that glass slide. They are probed with labeled mRNA from different sources. Active/inactivated genes are identified. So in your text, the scientific thinking shows this, and I'll kind of go through it briefly. But from such, the conclusion was that some genes are expressed in both flowers. In other words, the hypothesis was flowers and leaves will express some of the same genes, and by using a microarray, it was found out that some genes are expressed in both flowers and leaves, but some genes are expressed in flowers but not expressed in leaves, or in leaves but not in flowers. And you can see that at number 4, using that microarray. RNA sequencing, or RNA-seq, refers to the high-throughput methods of sequencing cellular RNA. It provides a snapshot of all of the mRNA in a sample by directly sequencing cDNA. Chromosome immunoprecipitation, or ChIP-seq, uses antibodies to find the genomic location of proteins bound to DNA. It's used to map locations-bound transcription factors and analyze actual protein binding, not merely the location. Proteomics. Proteins are much more difficult to study than DNA because proteins must fold properly to function. Post-translational modifications may affect function, and alternative splicing means multiple proteins can be produced from a single gene. Proteomic techniques. Mass spectroscopy determines the charge-to-mass ratio of a molecule, which identifies a molecule. It can reveal post-translational modifications, and protein microarrays are used to study large numbers of proteins simultaneously. Antibodies to specific proteins placed on a chip sample containing proteins applied to the antibody chip. So if you look here, it kind of goes through the process of mass spectrometry. It's used to analyze proteins. So the proteins can be isolated from the cells or tissue and partially separated using gels or biochemical techniques. So you see the cells, the protein mixture, separated proteins, and then the digested peptides, and then the peptide mixture, the separation of peptides by chromatography, the electrospray ionization, the ion peptide, and even the fragment peptide to yield to the peptide. And that's how mass spectrometry works to analyze proteins. Bioinformatics and proteomics. Bioinformatics is the application of computer programming, mathematics, and modeling to the analysis of large sets of biological data. And I would say this may be that part that may interest some of you all most. Currently, computer methods can be used to compare a protein sequence with already identified protein sequences. Computer models attempt to predict the protein structure from amino acid sequence. And this is pretty amazing here. So new computer methods can quickly identify and characterize large numbers of proteins. New proteins are compared to existing proteins based on sequence. Structure can be inferred, although different amino acid sequences can result in similar structures. Inference of function from structure is more challenging. Knowledge of a protein's evolutionary history is helpful based on shared structures and functions of common ancestor. And to apply genomics, let's go. So creation of a synthetic cell. Chemically synthesized mycoplasma mycoids genome placed in a mycoplasma capricolum bacterium. Synthetic genome controlled growth of the cell. So yes, it's now synthetic. And the expanding impacts of genomics include disease diagnostics, identifying genetic abnormalities, forensic diagnostics, identifying victims by their remains, tracing bacteria or viruses using bioterrorism, and study of complex diseases, using that genome-wide association study, GWAS, looking for the genetic components of disease. And with this, there are ethical considerations when you think of genomics. Opportunities of genomics are powerful, but must be balanced against the social and ethical responsibility of the technology. In 1918, the Spanish influenza virus recreated. Helpful to study the virus, but many people are concerned the benefits are not worth the risks. And then with gene patents, so a myriad of genetics So Myriad Genetics patented 2 genes involved in breast cancer. So that's Myriad Genetics, that company, patented 2 genes involved in breast cancer, and they gave them exclusive rights to test for gene variants. And the Supreme Court of the United States then ruled that because Myriad had not invented the genes, could not patent their sequencing. So it's real. And one last thing I'll state is that owning your own patented gene